I'll just get started introducing myself. I'm Navona Gallegos, and I work with the New Mexico Healthy Soil Working Group. Um, the Healthy Soil Working Group is hosting this webinar, and we are a nonprofit organization that advocates for healthy soil. Our mission is to support land stewards in enacting the healthy soil principles. And that looks a lot of different ways. We help with applying for different grants. We help with one-on-one consultation around soil health. We do a lot of outreach around soil health, and we also have a lot of partnerships with other organizations and groups that are working in soil health in some different areas like larger landscape scale restoration and also agrivoltaics, so combining solar panels with growing. Those are just a few of the things that we do. And if you want to find out more, you can go to our website, which you might have interfaced with a little bit to get on this webinar. The website is nmhealthysoil.org, and maybe that can pop up in the chat at some point. Can I just get a thumbs up or something from someone to make sure my audio is good and you all can hear me well?
Yeah, sounds good, Navona.
Okay, super. Thanks.
Sounds
Appreciate
good.
it. Nice to see you, David. Um, so yeah, I'm Navona Gallegos, and my background in soil started with studying ecology in school, and I learned a little bit about soil there, but more of my soil background really came in afterwards when I started to dive into permaculture and get really fascinated by how we can make regenerative growing systems for food. And um, that led me to find Dr. Elaine Ingham's Soil Food Web School. And that is how I got really introduced to all of the soil food web and the ecology of soil, and realizing that the organisms are really the main players behind plant health and ecosystem health, and how above ground ecosystem health really is a mirror and an effect of below ground soil health. So this webinar is intended to be sort of a primer, an intro for the soil food web, critters, and microscopy. So most of these organisms are microscopic. We can't see them with our eyes. And so using a light microscope is how we're able to see these organisms. And it's a really fascinating practice to use a microscope. And it doesn't have to be super technical or super complicated. Um, some of the approach we use with microscopy is doing soil analyses that the Soil Food Web School teaches. And some of the folks in here, like Josh Waybright and a number of other folks, are familiar with this quantitative soil assessment where we look at biology and count how many different organisms there are and make some educated guesses about extrapolating from a sample how many organisms are in the soil and looking at the different types of organisms in the soil. And then we can also do qualitative assessments where we're just looking at soil and seeing, okay, we have some bacteria, we have some fungi, um, and those types of assessments can be really, really simple. And if you can get your hands on a microscope, which isn't too hard to do, it can be really, really fun to look at compost or look at soil and not just observe what's happening above ground in terms of plant health and what kinds of plants are growing, but also look below ground and see what kinds of organisms are growing and how that relates to your land care practices. Um, so we can approach it either way. And yeah, I'm just excited to talk about some of these organisms that show up in the soil. And so to start, I just want to do a brief intro to the why of soil ecology, soil biology, soil health, and why we're even talking about these critters. And this might be background for some, but I think it's always worth just starting at the um, first stage, which is why do we even care? So if we look at the image we've got here, we've got this squash plant and some grama grass, and then we have the roots and all of these different organisms that are partnering with the roots and interacting with them. Um, Mind you, they're not necessarily to scale. So I've had some people look at this tardigrade and think that it's a larva or something. This tardigrade is microscopic, but these are some of the creatures that are in there, and they're illustrated so that we can see them. Um, so what this squash plant is doing is it's photosynthesizing. And that's where this whole engine of soil relationship starts. So the squash plant is receiving photons from the sun, and it's using that light energy to break carbon dioxide from the atmosphere into different pieces. So it's taking in photons and it's also taking in carbon dioxide. And we know this as, our basic um, photosynthesis equation where plants take in carbon dioxide, they exhale oxygen, we inhale oxygen, we exhale carbon dioxide. It's this really beautiful reciprocal thing that's keeping life on the surface of Earth going, and we're stoked about it. Um, and so what plants are doing with the carbon that they keep, since they're letting the oxygen back out into the atmosphere, what they're doing with the carbon that they keep is they're keeping some of it in their bodies. And then most plants are exuding, which is to say they're releasing or oozing 40 to 70% of the carbon that they create and that they take in through photosynthesis. They are exuding that into the soil around them. And nature isn't wasteful. So this begs the question, why is that carbon going out into the soil instead of staying in the plant? And the reason is because some of that carbon goes into making the cellulose and the different structures that make up the actual physical body of the plant. But just like us, plants need more than carbon to live. We have carbon in our bodies, right? And we have nitrogen in the form of proteins. But as you know, you can't survive on pure nitrogen or pure carbon. We need all kinds of other stuff, right? We need calcium for our bones. We need magnesium for our neurotransmitters in our brain. We need boron. We need cobalt. There's all these micronutrients um, that we require to make all the different things that make up our body and to allow our body to function. And it's the exact same for plants. So they require around 42 different nutrients in order to do their thing. And carbon comes in from the atmosphere, but everything else that they're getting from the soil. And so the brown background here on this image, that's the soil substrate. That is the sand, silt, or clay that the plant is growing in. And that is full of all kinds of different minerals. Now, those minerals are crystalline, and so they're not bioavailable. And so it requires the organisms, the bacteria and the fungi primarily, to make enzymes that can strip the atoms off of the rock that is the dirt in the soil. Because when we say soil, we're referring to three different things. We're referring to the dirt, the sand, silt, and the clay. We're referring to the organisms, the life forms in the soil. And then we're also referring to the organic matter, which is basically all the stuff that the organisms make. So like their poop and their pee and their dead bodies and the plant roots and everything else. Um, so the bacteria and the fungi are the ones who are the first generators of life in the soil. They're stripping atoms off of rocks and they're eating them. They're turning them into stuff, the stuff of their own bodies, their bacterial or fungal bodies. And then that ultimately becomes something that plants can eat. And sometimes that occurs directly, like we can see in this image down here on the lower left. There is a root, and we can see that that root has been infiltrated with a fungus. The little brown lines here are hyphae, and they're going in between the plant cell walls, and they're trading. So the plant is trading carbon, so carbohydrates or maybe proteins with the fungus, because the fungus can't photosynthesize. They have to get, just like us, they're heterotrophs. So they can't just get energy from the sun. They have to eat something. So they are getting those carbohydrates from the plant, and in exchange, they're trading whatever it is that they can produce. So maybe it's some kind of micronutrient like boron, for example. So that is happening right at the root surface. And you can see this green slime here is the exudate. This is what the plant is offering the fungus and maybe offering for other creatures. And as you can see in this image, there are all kinds of different relationships happening. There's a fungus over here, there's some bacteria down here, there's maybe some amoebas over here. And this is really what's going on the microscopic scale at plant roots, because a healthy plant will usually partner with a lot of different organisms. It'll partner with fungi, it'll partner with bacteria, um, and then it'll also partner with predators. Those predators are organisms that we're going to talk about in a little bit, and you can kind of think of them like the wolves or the cougars of the ecosystem, where the bacteria and the fungi are sort of like the deer or the elk that are eating the grass. They're taking the minerals from the soil, and they're also getting some things from the roots. Um, but then when they get eaten by predators, those nutrients then get deposited by the plant root as well and taken up. And so a plant actually does this interaction intentionally. So, when a plant needs to have more of a particular nutrient because, for example, they might be making seeds and they need some nitrogen to go to seed, then they're going to change the type of exudate that they make in order to attract, say, more bacteria that have more nitrogen in their bodies, for example. Or if they're flowering and they need maybe more potassium in order to create those flower structures, then they might exude something that will feed more of a fungus. And so this is happening all the time in healthy ecosystems and with healthy plants. And if we think about a forest, for example, no one's fertilizing that forest. That forest, all the plants are growing in symbiosis with each other and with the soil, and they're getting all of the nutrients that they need. So when we fertilize, we end up generally disturbing the soil ecosystem, and those soluble nutrients that the plants can take up that are in fertilizer are potentially really damaging to these soil organisms. And they also, the fertilizers that humans create, are not going to offer a full spectrum of the nutrients that a plant needs exactly when they need it. But in a healthy soil that has all of these different players, the plant can partner with whatever they need whenever they need it. So that'll shift day to day. And rather than us trying to guess what it is the plant needs, and if we just make sure that all of these different groups of organisms are present, then the plant can interact with those organisms whenever they need to and get whatever they need to. So that's really why we care so much about these infinitely fascinating, I think, microscopic creatures. Um, so we're gonna dive into talking about the different soil organisms, and these are functional groups of organisms. So in a healthy living soil, in a tablespoon of healthy living soil, there's more biodiversity than there is anywhere else on Earth, including the ocean. So this is an incredibly complex system. And this graphic is just attempting to demonstrate some of the interactions and some of the diversity that's under the ground. But it's a world that we really don't know that much about. And the light microscope was only came on the scene in, I think, the 80s. And so this is really a new and super exciting field, and we're discovering a lot all the time. And now with different DNA analysis, we're discovering even more about the different types of soil organisms that there are. But for our purposes, as maybe growers or enthusiasts, it's really important to just start with the different functional groups of organisms. And we don't have to get into every different type of bacteria because that would take a very long time, or every different type of fungus. But if we just look at the functional groups and know that there's some bacteria there, there's some fungi, there's some predators, then we know that our bases are covered. Um, and so that's where we're going to start. And as I'm going through, feel free to maybe pop some questions in, but we'll have more time for in-depth Q&A at the end. And so we're gonna just go through some of these organisms, and then hopefully we'll get Lina online and we will be able to real time look at some soil samples. So wish her luck. Um, so these are bacteria. Um, Bacteria are everywhere on Earth and underwater. Um, but if you wave your hand around, you're probably hitting some bacteria. You are covered in bacteria, which is a good thing. And the bacteria that we are usually looking for in a soil are aerobic bacteria. So these are bacteria that, like us, respire, which is to say they inhale oxygen, they exhale CO2, and in that process, they're doing a combustion process of that oxygen to create energy to fuel all of their functions. Um, so aerobic organisms. And these bacteria, there's a bunch of different types. We've got cocci, we've got lactobacillus, and um, actinobacteria. And if you notice, the main differences we're seeing here are their shapes. And so that's really what we're working with when we're looking under a microscope, is we're looking for the different shapes of the bacteria. And that's how we tell what's going on. So you've got the streptococci that make these little chains, um, Lactobacillus that have these little ovals, etc. And if we're seeing these guys, we know that we are looking at a soil or a compost that has had oxygen and is aerobic. Um, and the bacteria are, again, sort of our first players. And usually we're not lacking in bacteria. Um, but this is a little video that Lina took of some actinobacteria. And if we look at this video, you might notice all these little dots around here moving around a little bit. And these are all bacteria. So it's pretty, we usually, when we're looking at a soil sample or a compost sample, um, we're just seeing these little dots, and these are telling us that there's bacteria present. And they're also telling us that they're kind of giving us a sense of scale. You can measure the size of other organisms based on bacteria. So yeah, those are our bacterial friends. Is this the same one? Yeah, this is, excuse me. Okay. And
Hey,
then, oh,
Lina
excuse me.
is here, Navona. Lina
Oh,
is.
yay! Yay! You made it. Cool. Um, okay, feel free to pipe in if there's anything you want to mention.
And
So
to introduce yourself, Lina.
Hello. My camera won't start, so today I'm full of issues with technology. My bad. I hope I can still share my screen. Um, yeah, so should I go ahead and introduce myself? Okay, great. So my name is Lina Alegre, full name Carolina, you can see it there. And I am from South America, and So I'm a transplant, and I started getting into permaculture and about 10 years ago. And about 5 years ago, I met Navona, more like 7 years ago. I met Navona. Navona was already very interested in soil and pursuing the soil food web school and recommended it. And so, of course, I went for it and yeah, really fell in love with the magic of soil and really looking at it in the microscope gives it so much more aliveness to what we see. Soil can seem such a inanimate thing, and all of a sudden you see all these creatures and you're like, okay, great. So just because I can't see you doesn't mean you're there, which um, yeah, I really appreciate being able to see. Um, so hopefully I'll get to share my screen today.
Cool. Yay, I'm glad you made it.
Yeah, me too.
Um, so I'll keep cruising here. These bacteria are anaerobic bacteria. So instead of respiring, they do all kinds of other metabolisms where they might use sulfur or other compounds in order to create energy. And the it's not that all anaerobic organisms are pathogenic, but all pathogenic organisms are anaerobic. So organisms that are pathogenic to animals or plants are anaerobes. And to be clear, there are some anaerobes that are really beneficial. We see these in ferments like sauerkraut. Um, Things like lactobacillus can live in a low oxygen environment and be really um, helpful in providing nutrients for animals, for example. Um, but the anaerobic organisms, when we're looking under a microscope, we're usually not wanting to see these guys because they indicate that our soil or compost has low or no oxygen. And if that's the case, then seeing these indicates that there might be some kind of disease-causing organism. And if we're seeing one of them, that might not be a disease-causing organism, but it means that the conditions for disease-causing organisms exist. Um, and so we've got spirochetes, spirilla, vibrio, excuse me. And these have some interesting shapes and movement patterns. Um, so this is, excuse me, so this guy, the spirilla, which will kind of squiggle across a microscope. And over here we have a spirochete, which will corkscrew. And those are how they locomote. And so if we see these, we know that we've got low oxygen. And if we maybe see one, that's not necessarily an issue. But if we see a lot of them, then that is bad news. So the fungi, which are my personal favorite, um, do all kinds of stuff. They are those primary producers, which is to say that they can create enzymes that strip atoms off of rocks. And then they also do other things. So there are saprophytic, which is to say decomposer fungi that are taking things apart, all that organic matter that's in the soil. They're taking it apart and turning it into their bodies and other useful things. They're also creating a lot of the structure in the soil. Bacteria create some structure in the soil, but fungi do even more to create structure in the soil. And that structure is what allows water and oxygen to infiltrate the soil, which is really important for growing healthy plants, for having an aerobic environment and also a moist environment that allows plant roots to go deep and to be in a healthy microbial ecosystem. And so fungi, yeah, they decompose stuff, they help create structure. And then you've probably heard of mycorrhizal fungi that do the super fascinating and magical relationship between plants where they infiltrate the plant root, and they trade with the plant, and that helps get the plant. It's basically like extending the plant's root system, just as far as surface area goes, and it's also really increasing the plant's capacity to take up all kinds of nutrients, or water, for example, that they couldn't take up without the fungus. And the mycorrhizal fungi can also connect plants to each other, and so they mediate communication between plants. So fungi are just incredibly fascinating, I think. Um, and here we've got some fruiting bodies. Of course, not all fungi fruit as mushrooms, but some do. And so here we have a video of a fungus. One of the ways that we know this is a fungus is because we can see these septa, which are these sections that are here at regular intervals in the fungus. And then we also see how it breaks in a clean way at the end of a septum. And we also see some color in this fungus. We notice how thick it is in comparison to the bacteria around it. And we see that the edges of this fungus are pretty well defined. So all of those things are telling us that it's a fungus and that it's a beneficial fungus. There are some fungi that occur in lower oxygen environments, and those are going to be thinner. They're not going to have color, and they might have sort of wavy edges to them. So if we have a really beautiful strand like this, we're stoked. This is a photo of a fungus with a bunch of microaggregates around it. So all of this stuff is organic matter and dirt that's being glued together by the compounds that the fungus makes. And so you can see how the fungus is really tying together the soil. Okay, so now we're getting into the predators, and protozoa are a type of single-celled creature that eat bacteria, and so this helps cycle nutrients. And they usually do this. The bacteria will usually be near the plant, near the root, and then a protozoa will swim up and eat some of that bacteria and poop it out and make those nutrients available for the plant. And we have to have the predators in order for the nutrients to be cycled. We can't have a soil that's missing any of these different groups. If there's no bacteria, if there's no fungi, if there's no predators, then it won't work. We need all of the players. So here are a couple different ones. And again, these different predators occur in different oxygen environments. And we tell them apart based on the way they look, their morphology under the microscope. So let's see which one we got here. Okay, so this is a flagellate. You see the movement here, and you see how it's sort of bumbling around. So that flagellate, which is the organism that we saw on the last slide that has a tail, is moving around and eating bacteria in this slide. And so if it's not totally clear, because it's not still, we can at least tell from the way that it's moving what it is. These are some other protozoa. There's naked amoeba, there's testate amoeba, which are really fun. They make these shells made of silica. And if you get really close up, there's, with probably equipment that's higher magnification than what we're usually using to look at samples, their shells are really ornate. There's our cella. So these, again, eat bacteria and help cycle nutrients. This is a photo of a testate amoeba right there in the middle. So that's that silicone shell that it lives in, and it's inside of the test. Sometimes we see them with their little foot, their pseudopod, outside of the test, and you can even see them eating. You can see them full of bacteria sometimes. But this is what they look like under a microscope, and it's nice because they're usually pretty easy to spot. They have a pretty distinct look compared to a lot of the other organisms around them. This is a stellate amoeba right here in the middle. And you can kind of see some of the limbs coming off of it. And yeah. Okay, and so these are nematodes. They are, I was just checking the time. So nematodes are another predator, and they are really charismatic when you see them under a microscope if they're alive. They're pretty fun. They wriggle around, and I feel like they're a lot like snakes because sometimes they'll eat a big meal and then they'll slow down, or they can just whip around. Um, but yeah, there's different types of nematodes. I think nematodes sometimes get a bad rap in the gardening scene because of root feeders, but it's important to know that some root feeders are actually switchers. Some will eat roots if there isn't anything else in the soil for them to eat. But if there is fungi in the soil, then the root feeders will eat the fungi and help cycle nutrients from the fungi. So they can be beneficial or potentially detrimental. Um, at any rate, we tell them apart by their mouth parts because they eat different things, and so they have different apparati for doing what they do. So we can see this um, mouth part for the bacterial feeder. It crushes bacteria once it's sucked it into its mouth. The root feeder here has a spear. Um, let's see, the fungal feeder also has a little spear for stabbing fungi. And actually, this one, I misspoke. The root feeder has what we would equate more like a tooth, where the fungal feeder has more of a spear. And then this is an omnivore. It's got a big old mouth, and sometimes they have ornate lips. And then this predator, and the predator is going to be eating other nematodes, and um, we are excited to see predators in the soil. And so here's a bacterial feeder moving around. This is kind of a longer video, but we'll just skip around. Often we see these guys moving, and then the trick is to be able to focus enough to see the mouth parts. But it's always, I think, really exciting to see the movement. And we can even see some of the food that's inside of them. Um, so here's that bacterial feeder again. And once Lina gets on the screen, we'll be able to cruise around and talk about looking for nematodes. Um, so this is a still of that bacterial feeder. And here Lina is zooming in so that she can check out what's going on with the mouth. And so, yeah, I think we can see that this is a bacterial feeder. Um, It's not super defined, but we're also not seeing any teeth or lips or anything like that. Um, you know, this is a root feeder thrashing around here. I'm just gonna skip through the video a little bit more so you can see. So now so that last little bit, she was less magnified and just saw the nematode, and now she's getting more magnified so she can zoom in and try to figure out what kind of nematode it is. And there's just a second ago, you could see the little spear part there. Um, let's see. There's definitely some nuance to being able to catch these guys. And there's a trick where you can heat up the slide if you run a lighter underneath it and it gets warmer. You can overdo it and potentially kill the nematode, but if you just heat it up enough, it'll chill out, which is what we saw in the last slide. That one was after getting warmed up. So right now we're just seeing this one, which you can maybe take my word for it that it's a root feeder. Or fungal feeder, excuse me. I misspoke. It's a fungal feeder cruising around here. And then here is a close-up picture of that fungal feeder. Okay, so then we're getting into microarthropods, and these are basically tiny insects. Springtails and rotifers, for example. They are also helping to cycle nutrients in the soil. Let's see. This is a rotifer moving around. It's a little bit blurry, but it's kind of exciting. We don't see a lot of rotifers very often. So, and as you can see, working with the microscope is, yeah, this process of just getting to know how things move, what they look like, and being able to catch them long enough to figure out a little more of what they are. This is a picture of a rotifer. You can see they do this really cool thing where their mouthpieces spin around and they create a vortex and suck bacteria into them. So cysts are something else that we often see under the microscope, and they are protozoa or nematodes that have encysted, which is to say become cysts because the conditions in the soil are not favorable for them. So if we're seeing a lot of cysts, but we're not seeing a lot of protozoa or nematodes, then that tells us that the soil might have gotten a little dry or it might have gotten a little low in oxygen, or maybe there's a temperature fluctuation that caused those organisms to go into this protective shell. You can see that there's, you can see the edge of the shell. There's usually a clear edge here and a clear edge in here. And they can survive for a few months. These are soil mites, and now we're getting into organisms that are not microscopic, so it's possible to see mites. But since we're talking about the soil food web, it's worth just mentioning all the different players. So these are mites, and they hunt different microarthropods. They also can eat organic matter. And again, mites are something that sometimes have a bad rap in gardening, but are also crucial to the soil food web, depending on what kind of mite and what they're doing. Tardigrades are fun. They are microscopic, and they also help cycle nutrients, and they're just charismatic because they can survive all kinds of stuff and they have 6 legs. We don't see a lot of tardigrades often. I'm curious if any of the soil food web folks in here interact with tardigrades much, but yeah, I understand that they like more aquatic environments like mossy environments. So we don't see them much. Oh, I also want to just mention that a soil that is more than 30% water, which is relatively dry, unless a soil is really, really, really parched, then it's an aquatic environment. So because water sticks to things, it sticks to all the surfaces in the soil. So the water is sticking to the sand, silt, the clay, the organic matter. And so all these organisms are getting around by swimming. So our samples are definitely aquatic because we put water into our samples so that we can dilute it so that we can see the organisms. But even before we dilute it, it's an aquatic environment. And then these are some of the big critters that we're familiar with. Of course, earthworms. Earthworms are like taxi cabs, and they move through the soil. Their slimy surface carries a lot of the other creatures like bacteria and fungi around in the soil. They also have a lot of beneficial organisms like bacteria and fungi, aerobic bacteria and fungi in their guts. And so they're feeding them as they eat. And they're also, and in fact, the worms are not actually eating the organic material that they decompose. So sometimes we think of feeding worms like by putting food scraps into a compost, but actually they are taking up those food scraps. But what they're really eating is the bacteria and the fungi. They're squishing the bacteria and the fungi that are on those food scraps, and they're eating that. But a lot of the bacteria and fungi, especially the beneficial organisms, make it through um, and come out the other side of the worm. And pathogenic organisms do not make it through. So worms are wonderful to have in our compost, in our soil, and they are aerobic organisms as well. And then there's all the above ground organisms like birds and like us. So, Lena, are you set?
I believe so.
Cool.
I'll just make well, I wanted to ask, should we start with more, el, I'm gonna start with the El Rito, or I have some compost. Do we want to do El Rito or do we want to do just compost?
I think we should do
A comparison?
El Rito real quick,
Yeah.
just for like a quick visual, and then we can go into the compost. And I just saw that Josh mentioned that he found some, I think that's tardigrades on his Christmas tree lichen. That's cool.
Yep.
All right, I'll stop sharing and you can take over.
So this sample is soil sample from El Rito, which is where I'm from. And
I'm not seeing your screen.
okay, yeah, I'm just talking right now. I'm
Okay,
getting
sorry.
my sample ready because I don't want to make it too ahead for time because the samples do dry. Especially here in New Mexico, especially in a little room like I am with a wood stove, things will dry up really quickly. So let's see. Oh, I'm not on to view. Wait, stop share. I was ready on my other computer, but now I had to change. So anyways, we're gonna look at the the soil, and it's just soil, and the compost is aged compost, which I just found out today. And so that's a little less interesting of a compost to look at, but still very much um, different than just soil. So I'm gonna now share my screen. Okay. And we're trying to get people to get accustomed to looking at soil, right? Um, this might not be as diluted as needed, but we can still go ahead. So this is very clay soil. Can you still see my screen?
Yeah.
Okay, great. Um, Here it is. So this is very clay soil, not worked at all. So it's just mulched though. I do try to mulch my everything that I don't like bare soil, so I will mulch. And I've been living here for 10 years, so I've had time to do that. Um, so yeah, big overview. Nothing is wiggling or moving. So once you've done this enough times, you kind of go through it and you see what catches your eye, basically. And because we are trained to do that, right? We're trained to see, okay, what is moving or looking like fungi, or obviously I'm not looking for actinobacteria right now because they're thin, but I'm gonna look at that and be like, what is that? And so I'm gonna magnify and be like, is that fungi? What is it? Um, so can you see my cursor?
Yeah.
Okay, great. So this kind of it's dark in color. It's uniform. I think I see some septa, but I'm gonna keep. So what I'm doing right now is moving through the depth of the sample. Um, and so that gives me a good more depth and contrast, right? And I can't really tell, but yeah, so this tells me that it's not a fungi because it doesn't have that rectangular end, that 90-degree end. This more looks like a leg or something like that, potentially. And we don't count it, so it's like, okay, I don't, so I go back to my um lesser magnified, bigger bird's eye view of the sample and continue moving through my sample and seeing if there's anything that catches my eye. This is not for, obviously, I'm not doing this to count anything. I'm just showing a little bit of like what native soil can look like. This could have been more diluted, but for this purpose, works. And I'm not, now I'm seeing some sand, but sand is really rare. What I'm seeing more is clay. So the sand is really easy to see because, light moves through it and it creates this colors. This one right here. And it's jagged, right? It's jagged in shape. So that tells me here's another, sand particle. This is not a sandy sample, period. Like that's not enough. This is way more clay, which is information. It's information about your soil, which is it's valuable, I think. So nothing is really moving. I'm not surprised, right? There were no roots. Um, it was mulched, but that was it. And nothing was growing on it. It wasn't. So this is um, pre-inert, meaning the uh, oh the sand, the clay, the particles are there, but there's no life to it. So there's nothing to glue, let's say, nothing to glue these particles together, which is through microbial living. And so that is an important thing. Is like we're not seeing any big microaggregates, right? We're not seeing clumps of anything. So this is soil that can be easily eroded, for example, because it doesn't have aeration. It doesn't have ways for the water to move through it, that it has that weakness in it, right? And basically is the microorganisms that create those spaces. And my guess would be that that's a microplastic for its color, purple or blue. So I see someone actually moving on top. I don't know if it's moving or if it's just floating. And here too, um, let's see if I can find that real quick and see if we find any life. I would like to see some life. Um, and of course there is, but it's neglectable basically. And let's see. And it's hard to see because again, I didn't dilute it all the way needed. But just keep this in mind. They're not big aggregates in it. So that's gonna be the big difference. Um, and those are the things that are floating, they're just floating in the water because this is diluted, right? This is a sample in water. So things are floating around, moving a little bit, but it's not any microbes that I'm seeing. Um, all right, I think enough with this, and we're gonna move to compost. Oh, do we have until 6:30? Is that correct?
Well, we could go a
Yeah.
little longer since
Okay,
we had a rocky start, so it's all good.
great.
Yeah. Lena, I was wondering if it's maybe also because it's winter that there's fewer organisms.
Yes, and the same with the compost that I got. We're not gonna see, oh my gosh, it's like teeming with life. Besides, it's aged, but yes, winter is definitely a thing. But I wasn't seeing a lot of cysts or things, dormant, dormant shapes of things. But that's doesn't mean that they're not just sleeping somewhere else. Um, but cis is a good way to be like, oh, they were here and now they're not, but they're sleeping. They'll be back. That's a good sign. And if anybody has any questions in between samples, feel free to ask.
And this is kind of what we see a lot when we look at samples from like pinyon juniper, grassland, sort of bare dirt.
Yep. Yeah, not a lot of life. It's very um kind of sad and boring to look at, but you have to do it in order to like make a case and make a point to work with, whomever, the farmer, the gardener. Um, so now, oops, this is a compost that's aged 5 years. I had no idea. And um it was a Johnson Sioux that took some turns. So Johnson Sioux is a highly fungal compost, and that is sits for 60, 60 weeks. And um, it's supposed to eventually have a lot of fungi. The thing is, when you let it sit for 5 years and just water it, keep it water, the microbes do go back down in numbers because there's not enough food, right? Compost is just there's a lot of food, so there's a lot of microbes. There's a lot of habitat. There's just like they want to be there. Um, so this still is really good compost. Um, let's see. I'm gonna look into that just and see that's okay. That's nothing that we count, but it could have been like a huge actino strand. But now we see, and this is a 1:8 dilution, and the sample previous to this one, it was 1:5. So it wasn't a huge difference. And there's more microaggregates joined together. So they're not all over the place, all spread out, but they're actually joining together. And that is what creates structure and creates all the aeration in the soil, which is what we want. So here we can see a testate amoeba, um, I believe. Let's see. Ooh. And also there's like a fun little bacteria up there doing its thing. Sorry, I'm getting distracted. So the testate amoeba is this one, right? And then this is a bacteria of sorts. It kind of looks like a
What?
spirilla.
Yeah.
Yeah. So that's an anaerobic bacteria, and that's fun to see. Here's something else happening. So this it's just compost. It's aged, but it's still has a lot going on, and uh, um. It's moving in a way that's making me think. Otherwise, I would say maybe, no, I don't know. I don't know what that is. And this work is a lot of, I don't know what that is. This might be a spirilla, which is crazy. So that's awesome. and not great for the sample, but great for us to look at right now. And that's the thing. Compost is cultivating a whole bunch of microbes, a whole bunch of them, and we tried, to cater for the ones that are going to be beneficial for food production and for humans. And I would like to see if Josh has any thoughts on this one, if it's spirilla, if he can say yay or nay to that. So yeah, but this is a testate amoeba, I believe. And the entrance is here, and they do create these tests when there is alterations in the context. They just go. They're so vulnerable that they have this superpower of protecting themselves because they're really. And I think that this is another testate amoeba, if I'm. And yeah, so here's another bacteria that's an anaerobic. I'm pretty sure this is what's going on. And now that I'm seeing it here, because it was kind of hiding here and now it's moving. Modal, it's modal. That means, I mean, it just is what it means. And but also the big, the fast movement also tells me that they're anaerobic. They move really fast in low oxygen conditions, which is where I could have been keeping my compost in a low oxygen environment to keep it humid as well. So it's a give and take that you have to do. Um, I'm gonna keep going. Let's see.
I
Where was I
think
going?
Josh
This
wanted
way.
to chime in, Lina.
Yes?
Oh
And
yeah, I think spirochete, the
Uh,
bacteria.
Spirochete. Gotcha. Thank you. Um, that's
Yeah,
awesome.
it's you don't see them very often. It's pretty cool.
Right?
We had enough resolution we could actually see it. Good job.
Totally,
And
totally. Yeah.
I mean, it's an anaerobe. You don't really want to see them, but
Right.
it's cool too nonetheless.
Exactly. Yeah. I don't think I think I saw a spirochete recently. And now this is my second time. So, um, that's great. Here is a spore, a fungal spore, and they look very brown and they're pretty big. And this is one of their shapes. So that's something that we don't count for in our samples, but we do take notes and be like, oh, okay, well, that fungi went to sleep. That doesn't mean we can't count it as active, but we also take note that that's a thing. So here somebody showed up and it's bumbling and it's not really, I think it's a modal bacteria that came in. Um, so I'm just gonna keep moving. So again, this is sand. You can tell by, how the light is hitting it and also how jagged it is in the corners. This, on the other hand, is more plant material, organic matter. We don't count that. And we just we notice what color is it. This is gives a more fulvic acid color. This is a honey color. That means that there's fulvic acids in the soil, or in the compost, excuse me, and that's a good indicator that nutrients are being cycled. Um, so that movement was just me touching the microscope. So these things are uh, very sensitive. Um, I'm actually gonna go to 10 and just look around uh and see if I find anything, if we find anything fun. So, we didn't go too far. This seems to be, well, I can't say right now. I need to look a little closer. Yeah, it is a fungi. It's actually a huge fungi. Um, so why I know so sure is because of the septa. So right here we have one septa, two, three, four. they're there, hard to miss. And let's see if we can see the end of it. But yeah, it's got a nice tan color. That's another indicator of good beneficial fungi. Let's see. We keep going, and then the end is very square. Right here, it has a branch that comes this other way that I can also try to see. See, now I can. Oops. So the samples do move a little bit. So that branch, I can look at it and we count all of that length of that fungi is something that we look at, we count, um, and it's really fun. Once you learn that, but you don't need to know that. You don't need to know the how the length or the diameter gets accounted for. You just need to know this is a fungi and why. That's a very good example of like a beneficial fungus. Um, any questions with that? This one?
Yeah, so Valerie was asking if we're really rocking our garden and or farm soil health, would we commonly find protozoa and nematodes in soil here in central New Mexico in warmer weather?
You mean right now or just in general?
In general, and in warmer weather, and maybe more in central New Mexico, you're really northern New Mexico, right?
Without doing anything with your soil or she's saying she's working the garden, right?
Yeah, she's rocking her soil health.
Excellent. I would say yes. I would say she would definitely be looking at some life. Um, for sure. Yeah, yeah. If you're working your soil, then these beings want to be in your soil. They're not special to any another part of the world or anything like that. They are native to Earth and they want to exist. They want to live. They even go into cis mode in order to like continue surviving under not great conditions. It's amazing what they do, and if you give them the love and attention and care that they need, and microbes are kind of they're the same as plants. They need good habitat. They need water. They don't need so much sunlight like the plants, but you know what I mean. They're a lot like us basically. So here's a fungal spore in a different form. So I show you one kind of spore. This is another kind of spore. And these are really good. This is a good indicator of fungal activity.
Oh, that's cool.
Yeah, it's nice and dark. It's got that fungy uh, funk. Did I answer Valerie her her question? I wanna. Here's another fungi that got broken up most likely. Um, but yeah, so here it is, the fungal little strand. But it's actually really nice diameter. So this is a very nice, healthy fungi that's been growing nicely. And here's a fungal spore right here. And here's a testate amoeba. Um, and these are microaggregates that are mostly have fulvic acid. They're fulvic acid. And that's the honey color colors. And we can also see some fulvic acid microaggregates that um a little darker in color. Um, I'm gonna keep moving on to see if we can find any predators. Um, here's probably more fungi. I'm not gonna, we already saw a decent amount of it. More fungi here, most likely. Don't quote me on anything because now I'm not magnifying it, but I'm just cruising around. And so this is the really cool thing about microscopy is that you can your look at under the microscope. So see my okay, no. Look under the microscope. My sample is drying, so I'm gonna move to the other side. Um, you can just peruse and see more or less without having to know too much what's going on with your soil, which I think is such an advantage. Um, and I do reports. I'm a certified lab tech for the health for soil food web school, but so I do reports, and I find them useful. But mostly, I love the all the knowledge of these creatures, and sometimes these reports don't really tell the full story. So I will give a report that showed almost no fungi, but then I'll do this bigger looking around and seeing what there is, and I will see some fungi, and I'll be like, okay, I'm gonna put some notes and say there was some fungi, because if there wasn't some fungi, then that's notable, and we I need to know that. Um, so that's and this is it's not rocket science, is what I'm saying. Um, So I just saw something bumbling and I was wondering if it was like a flagellate, but you do need to find it once um once you see it, and I center it, put it in the center and see if I can find it sometimes with no luck. And there it is. But just for how it moves, I would say it is a flagellate. Um, but I can't say for sure until I look at it. Um,
While you're looking, do you want to say a word or two about the equipment that you're using and the
yes.
magnification?
Okay, so I'm moving mostly let's go. Let's see. So I'm moving from I'm using an Omax microscope with a BioVid 4K camera. So the Omax is the entryway microscope, nothing fancy. Um, it's fairly cheap, and the camera I think it's a little more expensive. Thank you, Navona and Isabel, for lending it. Um, so yeah, and then so I went from 4x just now, so I'm gonna do that again. So I'm gonna from 4x magnification, which means 40 magnification total, because it's 4 for the little piece. What is it called? The little pieces, and then the eyepiece is 10x always. The eyepiece never changes. So I'm going from 4x, which I can see a lot, and I'm gonna try to, there it is. Um, you can see a lot more, but then you can't identify anything very specifically, right? But you can see what's going on. Aggregates, no aggregates, fulvic acid, humic acid, no acids. And if there's a nematode, a rotifer, you're gonna see those right away. So that's something really, to me, it's neat. I like for X because I get to see this. Um, and be like, peruse through a sample really quickly and see what's going on. Um, and when I did this earlier, getting ready for this webinar, I saw nematodes, rotifer. So I'm hoping I'm gonna find some of those to show you. Um, yeah, the equipment is at least the microscope is a good way to, it's a good toy. It's a good toy. I like it.
And
I
you can
put
do this without the camera. You can just have the microscope. Um, but then if you're going to do it relatively often, it's pretty nice to have a camera so that you can look on a screen. Otherwise, the microscope can get a little hard on your eyeballs.
Yeah, that's one thing, and the other thing is the ability to do what we're doing, sharing it with other with people, right? So we did this thing at the Santa Fe Farmers Market where we came and showed, asked people to bring their soil samples, compost samples, whatever they wanted, and I'm gonna get another compost drop in here to see if we can see anything else.
Yeah, we
Um,
want to see a
or
Redditor.
You want to see
I
a
know.
radio?
No, they
Yeah.
all came out when nobody was looking, and then they got all shy. They were like, "Oh."
Oh,
Um, yeah, so we were at the Santa Fe Farmers Market and we were showing what, how soil looks like. And I think it's important to just like demystify soil and demystify like using a microscope, for example. And so, because again, it's not rocket science. Anybody can do it. And it's a matter of, do you want to, do you have the inner interest to do it? That's it. Um, so um it was really cool to be able to share with people right there their own samples, their own soils. It was really cool. So I really liked doing that and um yeah, I'm getting my sample ready. Any questions that are raising?
I think everybody's just enthralled.
Oh my gosh. Um, so this is a sample that's settling. So it's a sample that hasn't settled yet. Um, and so that's why it's got so many moving pieces. So again, we're gonna peruse through and see. So that, okay, that's just disregard.
Here's another question.
Yep.
Can you see humic and fulvic acids? Are they like crystals?
Oh, crystals. Um, hmm.
So if I can
Yeah, please.
hop in. I think so before when Lena was mentioning that she saw some fulvic acids, um it was the color, the sort of honey color that is the clue as to what kind of humic substance or humic fulvic or omic that is. So you don't really see like individual crystals of those acids. That would be quite a bit smaller, but
Right.
you just see the pigmentation that comes from them. And if it's darker, then it's humic, and if it's a little bit lighter, like that honey color, then it's fulvic.
Yeah, so I just want to also look at the fact that everything is more consolidated, or that might not be the right word. It has separation, right? Whereas the first sample, it was just so everything was just it was covered. The slide was covered. Here we can see again there's a lot of aggregation, which is really great and very different from the first sample. Yeah, so soil will take up those acids, will take it up and keep them there, and that's it's really great to have fulvic and humic acids. Those are carbon chains that are very solid. They are very stable, and that's carbon that's stored in soil for 100 years, 150 years. That's just there hanging out, waiting to be used up, but not in a way that's volatile at all. So that's what soil has the capacity to do. It has the capacity to store carbon.
This is the safari part, right? Where we just
Yeah,
cruise
totally.
around.
And I'm like, well, I took a video of the rotifers and the nematodes, so you all can believe me. And in case they didn't want to come out, um, we could look at them. That was just a couple hours ago. That's funny, they're not coming out. Here's somebody. So here's a nematode thrashing around, and yeah, like I in order to get that, can you all see it? So it's right here. This is the nematode,
Yeah.
and it's yeah, it's
Oh,
just upset.
oh,
It's a wild creature. I love seeing nematodes. Look at how wild that thing is. Um, so in order for me to ID it, I would have to go down in or let's see, down up in magnification. So we can, I can go in and see, and there it is, just moving, I mean, just being alive, basically. Um, And if I wanted to identify it, I would have to get a lighter, which you all can tell me if you want me to do that, and basically tranquilize it, stun it, and with the heat, and then we can potentially identify it. One of the slides that Navona showed, one of the nematodes was incredibly still. That happened because it was stunted. um, or stunned, right? Stunned. So this is how nematodes usually look like, and so impossible to ID, right? Um, so maybe sometimes, if you're lucky, you'll find a sleeping nematode. Teenage nematodes usually are found sleeping. I haven't had that luck. They're always thrashing and moving around and doing this, which is really fun and easy to like see them. If you want me, I can show, I don't know, I can show also how to do the lighter thing, or we can just keep safaring and moving forward. But that's a nematode, that's a predator microbe, that's cycling nutrients. It's doing really, really great things. What do you want me to do? Should I continue? Should I move forward, or do you want me to stun it? Keep going?
I'm thinking about timing because we're over our hour. Maybe we want to, and Isabel or anyone else, please chime in. Maybe we want to check out your videos
Yeah.
earlier, and then if there's any other samples you want to show, we could do that.
Oh, yeah, yes. So I have a pond sample that turned out to be really fun, that they had rotifers in it. That was really interesting. So that was great. Let's see. Oh my gosh. I just realized that the videos are in the other. Okay, so I think I just want to see. Because sometimes they hang out, the big guys hang out in the edges. So this is kind of like the last reading. Um, and this is sort of like an edge right here. Uh, yep. These are oil stains for anybody who's wondering. Um, yeah, and right now it doesn't look like much movement, but once we get closer and there's somebody in there that's moving. Um, I don't know if you can see it. It's right here. Um,
Mm-hmm.
Hard to say exactly what it is at this magnification. Okay, we're gonna do a pond, which that is not my expertise, because we're into soil, right? So we don't. I do like looking into water for water quality. I found some blue-green algae in my water through working through seeing it in my microscope. And I had obviously had some clues that that was what was happening. But then I confirmed it, which was really cool. And that's something that pretty much anybody can do, and it feels very empowering. Um, I didn't have to spend hundreds on a sample. I was able to confirm my doubts, and do something about it and get a filter. Um, so, oh, here they are. Great. So yeah, this little things, which I think they're rotifers. They're really fun, basically. So that's and Josh, if you know what these are, I have better pictures. Okay, so this is anaerobic, you know, full on life. So I don't know that much of it, but Navona had this great idea of getting some anaerobic liquids to see what was going on. So yeah, so these guys they look like rotifers in the sense that they when I get closer to them, so I'm gonna try to get closer. They they yeah, and they they look like it. They look like they're kind of pumping like rotifers do. Mind you, it's a totally different rotifer than what we're used to seeing. And this thing, I just don't even know. This thing. It seems animated. I didn't make this is happening live, okay? Anybody wants to chime in on what this could be? I guess I'm very curious.
I I agree about the rotifers.
Yeah.
Um, that's probably some kind of a ciliate, I would guess.
Oh, this is what? This is a ciliate, you're saying?
Could be.
Huh.
I don't see any hairs. Fun. I'm
And
gonna
then I
get
don't know what
right.
else would explain that movement. That's pretty interesting.
Interesting.
You should record that.
Yeah, right.
Mhm.
What is this? Well, this is a pond in Arkansas. Um, and I went to the middle of the pond to get this. Because I wanted the most like life that I could find at the I wasn't just gonna get the shore. Um, so yeah, this and then okay, let's look at some rotifers here. Oh, they were on this side, I guess. They're hard to catch. That's not.
Yeah, it's pretty weird seeing those together because the rotifers are multicellular organisms, which are usually very big, and those are particularly small ones. And ciliates are single cell, which are usually pretty small, and that's a particularly large one. So kind of crazy.
It is crazy, but it's also a pond, right? So as soil people, I feel like we don't see these samples as much. Is that it?
Mm-hmm.
I
Yeah,
feel like,
there's a lot of overlap, especially if you're in,
right?
in our drier climate. We don't have a lot of the,
Right.
you don't see a lot of these, even rotifers in soil samples very often, sometimes in
Right.
compost. But
Ah.
Yeah, you see them in pond water a lot. I see them in my aquaponics system.
Oh, excellent. Gotcha. Exactly. So yeah, yeah. Thank you for joining the conversation, Josh. Appreciate you. So now I'm just going up and down in magnification according to what we're seeing. So, um, so I can zoom in or zoom out accordingly. Um, that was a cute bump. Um, yeah, any questions coming up or yeah, this those bumbly things. This is my first time seeing this. Do you see these in, no, you don't see these in your aquaponics, you said. Um, I'll take a snap.
Oh yeah, and Josh just put this in the comments, and since we could keep on for a minute, but as people are starting to log off, just wanted to mention that we are doing this field day at Synergia Ranch on December 13th, and you can go to our website and look up events to find that and sign up for it. And we are going to have a bunch of microscopes that Josh is bringing. So you get to do this live and in person yourself if you're interested. Please join for that if you'd like. Oh,
Yeah,
great. There's the sign up right there.
Yeah, I just put the link to the field day in the chat. So that's just south of Santa Fe. That will be a lot of fun. We'll have lots of microscopes for folks to participate.
there's a fun little buddy there. Um, which I just lost. So
Yeah. Yeah, so maybe we'll wrap it up and we'll post the recording, of course.
And yeah, are there any more questions about any of this? Doesn't look like it. Cool.
Great job, you guys.
Thanks. Glad you're here, Josh. Thanks for going into the middle of a pond for us, Lena.
yeah, yeah, no, the things that we do to, have fun in the microscopes and,
Mhm.
just like my drive just now to in the snow to go get a computer that actually works and it's like, yeah, all for microbes pretty much and to spread the word that they're there just because we don't see them, they're there.
Yeah, lots of thank yous in the chat and thanks for making the trip through the snow.
My
And
pleasure.
sorry for the tech trouble.
I don't know. I don't know what happened there, but I'm happy that I got to be here for this. And thank you so much for putting it together, Isabel and Navona. Always a pleasure.
Yeah, likewise to all of you. Thank you all so much for joining. Um, yeah, please feel free to stay tuned. Check out our blogs as well. We've got some posts about soil organisms. A good one from Josh. That's a good 101 about all these creatures too, if you want to go over that as well. And yeah, maybe we'll do more of these if there's a little if there's some demand. Feel free to write in if you're interested, and we could take some more looks under the microscope in the wintertime. It's kind of fun since we might want to be indoors more to take this time to do more online stuff. Yay.
All right, well, let's log off. People are enthusiastic. That's great. Thanks everyone for joining and have a nice rest of your evening.
Bye.
Bye. Bye.